They are all around us, but we can't see them with the naked eye. Bacteria. It's only when we multiply them in the lab or examine them under the microscope that we can truly see their diversity. Over 1 billion bacteria can live in just 1 teaspoon of soil. Our own bodies contain around 2 kilograms of bacteria, with more than 10 billion in our mouth alone. Most of these are harmless. In fact, they actually help us, for example with digestion. Yet some bacteria can cause diseases and even become life-threatening. Whether in the human body or in nature, bacteria are masters of adaptation. They can survive even under the most inhospitable conditions. But they also have to defend themselves against constant competition. Some bacteria even arm themselves in this fight for survival. They build up a complex system of secretion that can shoot toxins in nearby bacteria or host cells. One type of bacteria equipped with this special nano-weapon is Pseudomonas aeruginosa. This bacterium lives in both soil and water, but it's also known as a multi-resistant hospital germ that can cause serious infections in humans.
In certain bacteria, we have what we call Type VI Secretion System. This is a nanomachine that assembles in the cytoplasm of certain types of bacteria. It takes about 10 to 20 seconds to assemble from one side of the cell to the other. And then, once this machine is assembled, there's a certain trigger that triggers contraction of this machine. And that's exactly the point where the proteins are being delivered from one bacterium to the other. And these proteins could be toxins that then kill the target cells, the neighboring cells. And in principle, this machine is sort of like a nano spear gun that allows bacteria to deliver toxins from one cell to another. Certain bacteria, such as Pseudomonas aeruginosa, assembles this Type VI secretion system in response to attack from neighboring bacteria. So they localize the machine and fire directly back at the attacking cells.
Until now, we did not understand how Pseudomonas sensed attacks from neighboring cells, but we had one assumption, which was that outer membrane damage actually triggered its response. To test this, we used the atomic force microscope, which is a nanotechnological device used typically to generate images of surfaces at the molecular level. So the AFM operates using a sharp tip that measures minuscule forces on surfaces. Our idea was to use the sharp tip of the atomic force microscope to mimic an attack while watching the response of the bacterial cells in real time using a fluorescence microscope.
First, we had to ensure that the bacteria are unable to move while we image them with the AFM. Therefore, we introduced a couple of mutations that fulfilled these requirements. We then set up the atomic force microscope to move up and down along a grid and indent the cells every 800 nanometers. In up to 90% of cases where we induced outer membrane damage to the Pseudomonas bacterium, it assembled this nano-harpoon within 10 seconds to fire back at the AFM. Interestingly, we observed that Pseudomonas counterattacks precisely in the direction where the attack is coming from.
Some bacteria have evolved a completely different strategy. Vibrio cholerae, for example, fires constantly, whether there's an attack or not. Both strategies have their advantages and disadvantages. For Vibrio, attack is the best defense, while Pseudomonas saves resources and only activates this nanomachine when necessary. We need high-tech equipment to explore such fascinating aspects of bacteria and other microorganisms. But it's worth it, because the more we understand them, the better we can use this knowledge to our own advantage.